Hello, welcome to my channel Deep Learning Biology. My name is Dr. Vikas Bhardwaj, and within this molecular biology class, I am going to teach you what is topology of DNA. So here within this video, you are going to learn what is topology of DNA, what is DNA supercoiling. Now here I just want to tell you, if you don't know what is topology, here I just want to tell you, topology, it is actually the branch of mathematics in which you study the properties of objects, which are not going to change under continuous deformation. So keep in mind, topology is actually the branch of mathematics in which we study the properties of objects which do not change under continuous deformation. Now here I just want to tell you, let us suppose you are going to have a ball, okay? This is a rubber ball, and if you are going to put pressure on to this ball, many of the properties of this ball, they are not going to change. All these properties of the ball which are not going to change under continuous deformation, they are known as topological properties. And what is topology? It is the branch of mathematics in which we study the properties of objects which actually do not change under continuous deformation. Now let us take here the example of DNA. Hopefully you know, in the case of DNA, what you are going to see two strands. The two strands of DNA, they are being intertwined to each other. Now let us suppose a protein is going to interact. So a DNA binding protein is going to interact with DNA. So here there is case of DNA and protein interaction. The protein is going to interact with DNA and it is going to lead to continuous deformation. So here the situation is of continuous deformation. So many of the properties of DNA, they are not going to change. So all these properties of DNA which are not going to change under continuous deformation, they are known as topological properties. Now here take another situation. In another situation, let's suppose you treat this DNA with a nuclease. Okay, the nuclease, it will go and bind to DNA, it will interact with DNA, and it is going to make cut within the DNA. So what is going to happen? Many of the properties of the DNA, they will change. Okay, so this is the example of discontinuous deformation. So here keep in mind, in one situation, many of the properties of DNA, they will change. So under the discontinuous deformation, we do not study the topological properties. So in this situation, the topological properties, they are undefined. But in the situation when we are going to have continuous deformation, the topological properties we can study. Hopefully it is clear to you what is topology and what is topological properties. Now here I just want to tell you, if you see the human cell, the size of the nucleus, it is 5 micrometer. Now if you see 2 meter DNA, in the case of human beings, 2 meter DNA, it has been packed within a small narrow space, within a small nucleus. Now here, if you see, you are going to have extreme compactation of DNA. So you see the length of DNA is 2 meter. The space is 5 micrometer, 5 into 10 to power minus 6 meter. So within a narrow space, 2 meter DNA, it has been packed. So here, what you are going to see, extreme compactation of DNA. Another important point here you must be knowing. In the case of DNA, you are going to have phosphodiester linkage. So within the phosphodiester linkage, you are going to have phosphoryl groups. And all these phosphoryl groups, what they do? They carry the negative charge. So when you are going to have large amount of negative charge in a narrow space, it will also destabilize the DNA. But here keep in mind, within the nucleus, DNA, it is being packed with magnesium ions, polyamides, histones proteins. All these, they neutralize the negative charge so that the 2 meter DNA, it is being packed within a narrow space. Yes, within this video, we are not going to discuss how the DNA, it is being compacted and it is going to form chromosomes. But here, if you see that many of the properties of DNA, they are not going to change under continuous deformation. So all those properties which are not going to change under continuous deformation are known as topological properties. So within this video, we are going to discuss the topological properties of DNA. Now here, if you see, if you are going to isolate DNA from the bacteria, let us suppose you are going to isolate plasmid DNA. You are going to isolate plasmid DNA from the bacteria. Now, you must be knowing in the bacteria, the plasmid, they are extra chromosomal DNA. They are circular in nature. And if you are going to isolate these plasmid, okay, so here let's suppose you isolate these plasmids. So one form of plasmid, what you are going to see is relaxed covalently closed circular DNA. Another form, what you can see that when you are isolating DNA, so you have added chemicals, okay, for the purpose of isolation of DNA. In that situation, the chemicals, they can break the DNA at random position. So what you are going to see, whenever you are going to have circular thing, if you break it at one point, it will get linearized. So what you are going to see, you are going to see linear DNA band. Next, within the cell, if you see within the bacteria, this DNA, it is being packed in such a way that here the DNA coils, they are going to make supercoils. So always remember, the coiling of coil, it is known as supercoil. So DNA, yes, it is going to have two strands, and both strands, they are being intertwined to each other. But further these coils, they are going to coiled another way, and these coils, they are going to form supercoiled DNA. So keep in mind the plasmid DNA, whenever you are going to isolate, in native forms, you are going to have supercoiled DNA, you are going to have linear DNA, you are going to have relaxed covalently closed circular DNA. All these forms you can see when you are going to separate this DNA via agarose gel electrophoresis. Now here, I just want to tell you, in the case of DNA, what you are going to see, you are going to see extreme compactation. Yes, the DNA, within the DNA, you are going to have two coils. These coils, they further coil one above other, and they lead to the formation of DNA supercoils. So here, keep in mind, coiling of a coil, it is known as supercoiling. Now, one important thing here, keep in mind, yes, here I have told you, you are going to have a very small space, 5 micrometer space. Within the 5 micrometer space, 2 meter DNA, it has been packed. But it has not been packed in a tightly fashion. Within the cell, all the process, transcription, DNA replication process, they are going to occur at a normal temperature. Both strands, they have to separate during the time of transcription, during the time of DNA replication. So here, the extreme compactation, the DNA supercoiling is going to occur in such a way that DNA, it is going to remain in underwound state, not within overwound state. How this is possible? This is possible that DNA supercoiling can occur in two ways. One, it can be in underwound state. Another, it can be in overwound state. Keep in mind, within the cell, DNA, it has been packed. You are going to see the supercoils, but all these supercoils, they are in underwound state, so that the transcription and DNA replication process, it can be carried out easily. Now here, I just want to give you the example. Let us suppose you are going to have covalently closed circular DNA. You are going to have covalently closed circular DNA, and it is going to have, let's suppose, 84 base pairs within the relaxed state. Now you must be knowing, within the relaxed state, one ton of DNA is going to have 10.5 base pairs. So 10.5 base pairs, they are going to make 1 ton of DNA. Hopefully you remember the structure of DNA. 10.5 base pairs, they are going to make 1 ton when the DNA is within the relaxed state. Now, if you are going to have 84 base pair covalently closed circular DNA, how many turns you are going to have? 8 turns. Okay, so here within this covalently closed circular DNA, 8 turns you are going to have. Okay, next, if you remove 1 turn, okay, now how many turns you are going to have? 7 turns. Okay, now if you see, you are going to have 84 base pair DNA, and now you are going to have 7 turns. Okay, in this situation, this 12 base pair per turn. So within 1 turn, you are going to have 12 base pair. So in this way, your DNA, it will be in a tension state. So DNA will be strained. Now, to overcome this strain, the DNA, okay, it will have one supercoil. Okay, so this is the supercoil. So here, by forming supercoils, in actually DNA, it is going to remove its tension. So hopefully it is clear to you what is supercoil. Now let us try to understand what is linking number. Linking number here, keep in mind, is actually a topological properties. I have told you, topological properties, they do not change. They do not change under continuous deformation. Now here, take the example. Let's suppose you are going to have double stranded circular DNA. Okay, in this double stranded circular DNA, how many times one strand it is being intertwined, it is being wrapped around another strand? This is known as linking number. Now take one example here. Let us suppose you are going to have a DNA. It is going to have 2100 base pairs. And if you want to know what is the linking number, you can calculate easily. Just divide it by 10.5. So here I have told you, within 1 ton of DNA, you are going to have 10.5 base pair. So if you divide 2100 divided by 10.5, it will come out 200. So this is the linking number. Hopefully it is very much easy. Now let us suppose you are going to have a DNA, covalently closed circular DNA, and if you are going to make cut within this DNA, okay, by using any nuclease, under this situation, keep in mind what you have created, discontinuous deformation. Under this situation, the linking number is undefined. Let us suppose you are going to have a covalently closed circular DNA where the linking number is 200. So LKO. Here it represents the linking number in the relaxed state. So let us suppose you are going to have a covalently closed circular DNA where the linking number is 200. And let us suppose you remove 2 turns. So you have removed the 2 turns, and next the linking number is now 198. So here you can easily calculate what is the change in the linking number. So LK minus LKO. So here it will come out 198 minus 200, that is minus 2. Now here, if you want to calculate another term, that is sigma, okay, it is super helical density. So here, if you calculate the sigma, it will come out minus 0.01%. Okay, minus 2 divided by 200, it will come out minus 0.01%. What does it mean? It means 1% of helical turns, 1% of helical turns, they have been removed from the DNA. So here keep in mind, sigma is actually the specific linking difference. You can easily calculate. It is equal to change in linking number divided by the linking number in the relaxed state. And if you calculate, it will come out with some percentage. In the first example, it will come out minus 2 divided by 200. And here it will come out sigma, that is minus 0.01%. Now in the case of cellular DNA, generally the value of sigma, the specific linking difference, it is equal to minus 0.05 to minus 0.07. What it means? Means 5 to 7% of helical turns, 5 to 7% of helical turns, they have been removed from the DNA. So keep in mind, within your cell, the DNA, it has been packed such that the 5 to 7% of helical turns they have been removed. Now let us have another situation. Let us suppose you are going to have covalently closed circular DNA. In this situation, let's suppose you increase the linking number by 1 unit. So here you are going to have covalently closed circular DNA, relaxed DNA. And if you increase the linking number by 1 unit, here within this DNA, you are going to see the formation of positive supercoil. So here if you see the supercoils, they are going to move in a right-handed manner. So if you see from bottom to top, so the coils, they are going to move in a right-handed direction. So the positive supercoils are being formed. They are right-handed. Now, if you see another situation, let's suppose here you remove one coil. Okay. So here you have changed the linking number by -1. Here also what you are going to see, you are going to see the formation of negative supercoil. So negative supercoil, what they are going to appear like? They are going to appear like left-handed. So here you are going to see positive supercoil and negative supercoil. Always keep in mind, negative supercoil and positive supercoil, they are mirror image of each other. And these DNA, which are going to differ in their topological properties, they are known as topoisomers. Keep in mind the DNA, here one is having negative supercoil, another is having positive supercoil, and both of the DNA, they are going to differ in their topological properties. They are known as topoisomers. Hopefully it is clear to you what is topology, what is topological properties, and what are topoisomers. Now here if you see the enzymes topoisomerase, the enzymes topoisomerase, they actually change the linking number of DNA. They can either increase the linking number, they can either decrease the linking number. So keep in mind, topoisomerase are the enzymes which actually change the linking number of the DNA. Now here I just want to give you an overview. We are not going to discuss in detail different type of topoisomerase. So here I just want to tell you topoisomerase, broadly they can be classified in two categories. One is type 1 topoisomerase, another is type 2 topoisomerase. Now here if you see the case of type 1 topoisomerase, the type 1 topoisomerase, they actually break one strand of DNA. So they are going to make cut in one strand of DNA. So here the type 1 topoisomerase, they are going to change the linking number by 1 unit. Here they relax the DNA by removing the negative supercoil. So keep in mind the type 1 topoisomerase, they break one strand of DNA, they change the linking number by 1 unit, and they relax the DNA by removing the negative supercoil. So here keep in mind the bacterial topoisomerase 1, bacterial topoisomerase 3, they both are example of type 1 topoisomerase. Next here I just want to tell you what are type 2 topoisomerase. Type 2 topoisomerase, they actually break both DNA strands. They are going to make cut within both strands of DNA. So at the time of DNA replication, when the DNA replication process is complete in bacteria, at the end, what you are going to see, you are going to see the formation of catenated circles. These circles, they must be separated. And here, the type 2 topoisomerase, they will make cut in both strands of DNA. And this way, what will happen? The one DNA, it will come out. Okay, this way, both strands, they will separate. So keep in mind, type 2 topoisomerase, they break both strands of DNA. Here, what you are going to see, you are going to see the change in linking number by 2 units. So here, keep in mind, the example is DNA gyrase. DNA gyrase, topoisomerase 2, is actually example of type 2 topoisomerase. By using ATP, it is going to introduce negative supercoil within the DNA. So hopefully you have learned different things, basic things related to topoisomerase. Now here I just want to tell you one classical example that during the time of DNA replication, what happens? The two strands of DNA, they separate. The two strands of DNA, they separate, and here this role is being done by the enzyme DNA helicase. Now here what you are going to see, you are going to see the formation of replication fork. Now here if you see, ahead of the replication fork, what you are going to see, you are going to see the compactation of DNA. So here what you are going to see, you are going to see the DNA, it is present in overwound state. When you are going to separate two strands, so at one end what you are going to see the overwound DNA, on the another end you are going to see the underwound DNA. This overwound DNA, it will create tension. So it will not allow the helicase enzyme to separate both strands more. Okay, so in this situation, what is going to happen? DNA gyrase, this enzyme, it will relax these supercoils, and this way it will introduce negative supercoil within the DNA. So here overwound DNA, it is going to have positive supercoils. These negative supercoils will be introduced. So here the negative supercoils will be introduced and DNA will become underwound. So in the underwound situation, cell, they can easily separate both strands of DNA. In the overwound situation, cell, they can't separate the both strands of DNA. So when the process of transcription is going to occur, when the process of DNA replication is going to occur, in that situation, if the two strands of DNA, they are overwound, what will happen? The cell, they can't separate both strands of DNA easily. So here, what is the strategy? The cell, they keep the DNA in the underwound state. So here, if you see, this DNA is in underwound state, and here, if the DNA become overwound, the cell, they have the enzyme topoisomerase, they can introduce negative supercoil within the DNA. This way, the cell, they maintain their DNA in the underwound state. Hopefully, you have learned basic things related to topology of DNA. Thank you for watching this. Have a nice time.